In the previous video, we covered the mechanisms of B and T cell activation. In this video, we will add context to these mechanisms by discussing the where and the when of the adaptive immune system activation. The process begins at the site of exposure. In the periphery, exposure to a pathogen activates dendritic cells. When activated, the dendritic cell expresses CCR7. CCR7 is the receptor for CCL21, a chemokine made by the HEV cells and other lymph node cells. The dendritic cells, with CCR7, therefore migrate to the lymph node, where they reside in the T cell area. Because they are activated, the dendritic cells express B7 on their surface. Meanwhile, naive T cells also express CCR7 and therefore migrate through the lymph node tissue. In the lymph node, the T cells interact with the dendritic cells in the T cell area. If there is a dendritic cell with an MHC class II which matches the T cell receptor, the T cell will bind and become activated. Notice that the dendritic cell also expresses the B7 which interacts with the CD28 on the T cell to result in full T cell activation. Within the lymph node, the CD4 T cells will proliferate and begin expressing chemotactic receptors, attracting them towards the cortex of the lymph node. Because these T cells are activated, they express CD40 ligand on their surface. We will pause here and discuss B cell activation. The pathway to B cell activation also begins with the macrophages and dendritic cells in the periphery. Activated macrophages and peripheral dendritic cells migrate to the lymph node cortex. Here, using CR2, they transfer the antigens to the follicular dendritic cells that reside in the cortex of the lymph nodes. These lymph node dendritic cells present the antigens on their surface, and B cells circulating in the lymphatic system sample the antigens presented on the dendritic cells for a match to their B cell receptor. When a match is found, a B cell endocytoses the receptor and proliferates. Some of the B cells differentiate into plasma cells and secrete IgM, while other cells express the endocytosed contents on MHC class II and begin migrating towards the medulla of the lymph node. At this point, notice that the B cells are migrating deeper into the lymph node, while the T cells are migrating towards the periphery. When they meet in the middle, the B cell will interact with any T cells containing a T cell receptor that binds to the fragments presented on the B cell MHC class II. At this point, the B cell CD40 interacts with the CD40 ligand on the activated T cells. With both the receptor signal and the coactivator, the B cell and T cell conjugate pair will migrate to the cortex together and begin the process of somatic hypermutation and class switching through the generation of a germinal center. Signaling factors expressed by the T cells and present in the milieu of the lymph node will determine the final class of the antibody produced by the B cell. As a reminder, during somatic hypermutation, the B cell receptors undergo random DNA mutations in an attempt to make higher affinity receptors. If a receptor mutates and has a stronger interaction with the antigen, the B cell will survive. If the receptor mutates and fails to bind to the antigen, the B cell will undergo apoptosis. The result of somatic hypermutation is high affinity receptors that is secreted as antibodies. Of note, the final form of B cells that secrete antibodies is referred to as a plasma cell and migrates to the medulla and into the lymph and peripheral circulation. To summarize, in this video we covered the sequence of events that takes place in the adaptive immune response and their location in the body.